at rest and during stressful stimuli, and we did their surveys regarding gastrointestinal symptomatology, and we looked at the heart rate variability from the EKG and the mean skin conductance from the GSR, and we asked the question how stress reactivity might relate to gastrointestinal problems and autism spectrum disorder. Let's let the slide shift. Can we have the next slide? I seem to have lost that ability. Okay, maybe I can control that thing myself. I'll see if I can do that. All right, so I think we skipped a couple slides here. At any rate, the next, there it is, the next slide. Okay, lost one of my printouts. So our hypothesis was that its heart rate variability would decrease, symptoms would increase, because a higher heart rate variability is actually associated with greater parasympathetic activity, and as you rev up a sympathetic response, it becomes more rapid and more regular. Skin conductance response is a more sympathetic marker, and it increases as gastrointestinal symptoms increase. That would be our hypothesis. Awesome. Now we're back to where we should be. So we looked at, and this is what your tracing would look like. Skin conductance is traced over time, and that's the red bar that you see there. And also the heart rate variability can be measured by following the EKG line that you can see. It's the blue line on the bottom, and and in 50 is the most typical heart rate variability measure. That's a percentage of normal RR intervals that differ by more than 50 milliseconds. And on our second aim, we examined endocrine markers and immune markers, and we took the same children from both sites, collected blood and saliva samples, examining IL-6 and TNF-alpha, and look to see if these co-segregated with gastrointestinal symptomatology among autism spectrum individuals, with the theory that immunological function would be greatest, reactivated the most in those with the greater stress reactivity, and salivary cortisol would increase with gastrointestinal symptoms. A little background on that aim is that there are a number of immune markers that have been reported as atypical in ASD, and several of these are also associated with gastrointestinal symptomatology. There's significant overlap between those markers and those that are associated with the stress response, in particular IL-6 and TNF-alpha, and so those were our primary targets in this. And we are also interested in the stress response as measured by cortisol. And the third aim, which Jeremy will talk about, I'm going to briefly introduce as well. We looked at whole blood serotonin levels and looked at how these interrelated with these other factors in autism. But I will defer that to Jeremy's talk, which will be the last half of this section. So the Rome-3 measures severity of functional gastrointestinal disorders. Again, as I mentioned, this is beginning to be used as a measure of autistic symptomatology for gastrointestinal problems. And it's typically used as a criteria to determine whether someone meets criteria for gastrointestinal disorder. However, we created a scoring algorithm to create a continuous variable as to the severity of the gastrointestinal impairment based upon this. We enrolled subjects aged 6 to 18, and we excluded those with a known mitochondrial genetic syndrome, sensitivity to adhesives, inability to remain still, bleeding disorder, medical conditions that might interfere with the problems, lack of a reliable informant to tell us about the symptoms, and obviously those who would not want to consent. The mean age of the subjects was 11.89, right in the middle of the age range, and 120 were recruited, and it was predominantly male to an even greater extent than typical, but that may reflect the numbers with the symptomatology. And when we looked at the heart rate variability, we found several interesting things. First, we found a fairly strong relationship towards increased heart rate variability with lower gastrointestinal symptomatology at baseline. That's the lower left square there. I highlighted the significant ones in green and the trends in yellow. There was also a significant relationship between lower in the middle bottom item there, the cold pressor. The cold pressor minus the baseline, so the difference between cold pressor and baseline, what effect cold pressure had on heart rate variability was also significant for the lower GI, and we had a trend for the same thing for vibrotactile stimuli. For the upper GI, we also had a trend for the baseline in 50 in the same direction. And for the cold pressor, there was a positive relationship, but it was only a trend for the cold pressor with upper gastrointestinal symptomatology, but it was only a trend. This summarizes the things that were significant. For the lower GI, there was a significant relationship both for the baseline NN50 and the cold pressor baseline difference NN50, and the others are trends. And we did a regression analysis as well, and this revealed that for the upper GI, the symptoms were predominantly driven by the NN50 baseline. That trend there was driving everything, and there was a small contribution from heart rate, but no heart rate effect reached significance in any comparison. For lower GI, it was predominantly driven by the cold pressor effect. However, I should comment that the cold pressor and the baseline NN50 were very closely interrelated for the lower GI, and so basically whichever one reached the greatest significance, which in this case was the cold pressor, was driving the overall regression. And the AIM-2 results are forthcoming. All the analyses have been, actually they've been finished, but are being analyzed and will be entered into the data set. So in short, the psychophysical markers, there appears to be a relationship between NN50 and lower gastrointestinal symptomatology, and perhaps a relationship to upper as well. The GSR findings are somewhat limited. These are technically challenging, and they're much more susceptible to motion artifact than NN50 and then the heart rate variability, and therefore the samples were not as clean. I would comment that the baseline and stimulated heart rate variability aspects are very closely interrelated, and I would suspect that what is going on there with those findings is that if somebody is maximally sympathetically stimulated, they may have reached a ceiling, and therefore this would impact how much change can occur with stimulation. And so I would, we're driving this with subsequent analysis. We're going to drive it based on parasympathetic being the obligate primary factor in the regression analysis, since that would seem the most appropriate given this setting. However, there is one last thing we do want to do as a post hoc frequency analysis, because we can't be completely sure how much of this sympathetic effect of the NN50 may be a reaction to some other sympathetic factors that are going on as well. And so a post hoc frequency analysis can look more closely at the sympathetic and parasympathetic components from heart rate variability, and that is being done right now. We also looked at relationships with behavior aspects as well, and the cytokines, again, as above, are completed and being explored. In the future, it certainly would be interesting to look at other factors, how stress susceptibility genetics relate, how the microbiome relates to these factors as well. And this may ultimately have an impact on treatment. We need to wait until we have this frequency analysis completed so that we have a little bit more of a definitive answer on the sympathetic versus parasympathetic component before we move forward with this. Because if the sympathetic component is driving this, it would certainly be of some relevance to to what you might want to do in a treatment approach. Because if it's sympathetic driving it, then anxiolytics may be something that would be an important component of treatment approaches for individuals with gastrointestinal disorder based upon this biomarker. And ultimately, that's the hope, is that this becomes a biomarker that can ultimately impact the treatment plan and begin to predict who is most likely to respond to standard treatment for gastrointestinal symptomatology and who may require another approach. A little ways to go yet before we get this, but this is the first study, to our knowledge, that has really begun to explore this question. And with that, actually, I ended a bit early. I usually do talk quickly, and thank you. I'd like to thank Brad Ferguson, who basically was the motor running behind this whole engine, and really is doing this as part of his PhD thesis. Jill Akers, she is our coordinator. She's the person that was affiliated with the Thompson Center Research Corps that was absolutely critical for getting this off the ground. Micah Masaryk helped a lot with the anxiety assessments that we've also begun to look at. Caitlin Hartnett and Brianna Killey helped considerably with the project itself, the Thompson Center, and also Evan Lee and Sarah Marler and obviously Jeremy at Vanderbilt, and the incredible statistical help we've gotten from Eric, Lily, and Aaron. And I will pull out here and let everybody pop in for a couple questions before Jeremy joins the line.
So we have a couple questions. So the first one is, how might the results have been biased by the exclusion of subjects who couldn't sit still?
That's actually a really good question, and we're going to have to explore the exclusionary group. We have not yet done that, to see how they differed from those who could. And also within the GSR group, I think we'll want to look at that as well, because a lot of them failed to be able to hold still for that. But that's actually a really good question. We don't know the answer to that yet.
Okay. And did anyone else have any questions they'd like to pose? You can either raise your hand or type them in the questions bar. We have a couple minutes before Jeremy can join us. Okay, so we have another question. Can you provide some more information about potential therapeutic benefits of the study?
Well, the hope is that if we can, the hope is that if we identify a sympathetic factor, that individuals that may fail to respond, the next wave actually I'd be really interested in is who does and does not respond to standard treatment. And so you can give, for upper gastrointestinal symptomatology, you can give a proton pump inhibitor or something else to try to settle down the tummy. But if anxiety is driving this, then you would certainly want to consider another treatment approach. So we can look at this. One of the next things I'd be really interested in doing is tracking how people have responded to standard treatment. And if our psychophysical markers tell us, whoa, wait a minute, the ones that have that really have bad sympathetic activity are not the ones that are going to respond well to standard treatment, that suggests that you may want to add on, say, an anxiolytic to the treatment approach for these patients. And that's kind of where we hope to go with this in the treatment direction.
Okay, great. And then how well did the Rome questionnaire work? And did it distinguish well between subjects without GI, with GI, and those clearly without GI?
Well, there's two answers to that. Number one is that the Rome questionnaire is co-segregated really, really closely with the survey answers on the ATN GI questionnaire. So if you reached criteria for GI on one, you reached it for the other. Although one might consider the Rome 3 to be more of a gold standard because it is a more detailed questionnaire in that regard, but it's nice that they corroborated quite well. However, the second answer to the question is we added a twist to the plot by making it a continuous measure and allowing us to improve our statistical ability to detect effects and that has that, of course, would just basically be fleshing out the data and creating a continuous score based on upper and lower gastrointestinal symptomatology rather than a dichotomous positive or negative reached criteria response. And so that would just be fleshing out the data provided by the dichotomous Rome the diagnostic questions.
Okay, and so again related to the Rome, what was the distribution of scores on the Rome, a broad distribution versus a separation?
That, I'll have to pull up the graph and I'll have to describe it to you because at one point had that slide, but it was very, very complicated, and we went away from that. Final push. So so the thing with that is we actually were trying to make sure, in all fairness, there's the range was impacted by our recruiting strategy. And so we were trying to make sure we had an even distribution of people with and without Rome 3 diagnoses. So what we ended up with is half of them having no Rome diagnosis and half of them having some degree of diagnosis. And let me look at the range. So it ended up being half and half had it. There we go. For the upper gastrointestinal symptomatology, the scores rate, or the mean scores were 11 with a standard deviation of 11. For lower, it was 16 with a standard deviation of 11. So it was ranging from 0 to 50 and 2 to 48 across all subjects for the upper and the lower. So it was a pretty wide range, and being that half of them did not have symptomatology, the mean was going to be weighted towards the lower end of the scores. And then the other half would be above criteria. So, so in order to get us an adequate range, we had half and half with and without.
Okay. And so then, do you have any comment on the role of beta blockers to damper sympathetic activity?
That's actually an interesting question because when we get the final answers for that's the nice thing about the design that we had in coordination with Jeremy, is we're going to ask the serotonin and the adrenergic questions. So if we find out that the sympathetic seems to be driving this when we do our frequency analysis, then one could propose that controlling adrenergic tone would be a good approach. And secondly, if we later find out that that patients with higher sympathetic reactivity are the ones that fail to respond to treatment, then the same answer would be a consideration because it's sympathetic activity that's the issue. The nice thing about Jeremy's study is that he's looking at the serotonin factor, and that would implicate possibly a serotonergic approach. So that is a possible outcome, but the last pieces of the question are still forthcoming before we can make that call. And of course, one would want to test it out in a trial.
Okay, and we have a couple more questions.
Sure.
What was the range of functional levels in your subjects? And then do you think there was any association between functional and verbal level and GI symptoms?
The mean IQ was 84 with a standard deviation of 23, ranging from 36 to 130. The nice thing is the demographics page was the first thing that popped up when I opened up the stats column, so that worked out rather well. As far as relationships with IQ, I don't recall that we found any there. I'm going to pull up that that spreadsheet and try to look for that. But I don't recall there being a relationship for those, but I will, give me a second here. ADOS. there was no relationship with IQ for symptomatology.
Okay, and so then how do these findings compare with typically developing children?
Well, that's actually, in order to focus the study, we definitely wanted to concentrate on autism, but we haven't systematically asked that question yet because everybody's methodology is slightly different, and the Rome-3 has actually not been used as much in this setting in typically developing children. It's a very perfectly reasonable question. We're targeting this in order to look for what can be done in autism because of the special features of behavioral worsening with gastrointestinal symptoms. So it has possibly a higher impact in autism. However, as to the effects in unaffected individuals, it may well be the same. It just doesn't have quite the same behavioral impact and quite the same incidence of gastrointestinal problems. But we haven't really addressed that, and the Rome-3 hasn't been used quite in this setting, so we don't really know for sure.
Okay, and did you query any other ANS symptoms in this?
Yeah, we looked at heart rate as well and found no relationships across the board. And now we're looking further at frequency analysis to subtype the sympathetic and parasympathetic factors in heart rate variability. But that's what we focused on in this study. But heart rate was also done but didn't reveal any relationships.
Okay, and do you know of any similar studies that have been done with functional abdominal pain or irritable bowel syndrome in typically developing kids, and if they compare with your findings, if any are available?
No, this is actually, the way we looked at this is kind of new because it appears we're the first to have developed or to derived the continuous variable for the Rome scores, and so I don't believe this has been done in other populations.
And maybe that can direct some future research.
Quite possibly.
So thank you so much, David. I think that we have Jeremy on the line now to present his subset of the data from this study. Jeremy?
Yes. Can you hear me?
Yes.
Fantastic. Okay. And we'll see if I can actually advance slides. Yes, great. Thank you for inviting me to present. I'm sorry I didn't get to hear David's presentation. It sounds interesting. Hopefully most of those data are already familiar. I'm going to spend some time setting the scene as to why we would be interested in looking at serotonin in relation to GI symptoms, and then talk about data from this particular analysis. I want to highlight funding and disclosures. I'm not going to talk about anything related to industry-sponsored work, but I would note that I've had industry funding for research studies. And there we go. So I think when I think about a child with autism, I never think about a child who only has autism. In the center, you see core autism spectrum disorder symptoms, but typically what we see when a child actually comes into the clinic is perhaps something that looks more like this, where a child also has language impairment, intellectual disability, perhaps a seizure disorder, and sometimes some biological markers related to autism or possibly related to autism. But when we think about autism spectrum disorder, we typically take that child and say this child, who sort of is the hypothesis of this study, that you would see a child with autism and perhaps more repetitive behaviors, might see a particular biomarker that I'll go into in some detail, and then might also see particular medical comorbidity that may identify a subgroup that could be amenable to a common treatment. So I'm going to talk about this potential biomarker, elevated blood serotonin levels. I'm going to talk about some work in the rodent that led us towards some of these hypotheses, and then talk about specific outcomes of this study. So when we think about this endophenotype, this is something that's actually been around for a long, long time. So in 1961, Daniel X. Friedman first identified elevated blood serotonin levels in roughly a quarter to a third of kids with autism spectrum disorder. If you see this graph here, in the typical population, you would see a bell curve tailing off here, but in the ASD population, you see in some studies a second bell curve sort of superimposed on the first. Depending on where you draw that line, you see that it's roughly a quarter to a third of children. The serotonin that is identified in blood studies is contained almost entirely in platelets. So this is a platelet. I don't know why there's a box in the middle of it. But the platelet you can think of as sort of a reduced system. It lacks a nucleus, and so you don't necessarily see a lot of transcriptional regulation. You see the platelet taking serotonin up as it passes through the gut circulation, packaging it in vesicles, releasing it in turn. There are some serotonin receptors on the platelet. But this gives us a picture of the sorts of genes and proteins that might be involved in handling serotonin. There's been a lot of work focusing on what might be changed at the level of the platelet in autism. But more recently, there's been interest in understanding the impact of where serotonin is produced in the gut. It's actually produced in enterochromaffin cells, is important for gut motility, hence the interest potentially in constipation, and is also important for regulating inflammation in the gut. The serotonin levels in the blood are actually more genetically controlled than is autism itself, pointing to this as a potentially useful biomarker. And serotonin levels in the blood are linked with the gene for the serotonin transporter, shown here, responsible for taking serotonin up into the platelet. And interestingly, in autism, you see strong evidence for genetic linkage overlying the serotonin transporter gene, only in families with males affected and no females affected, paralleling an association with this gene in individuals with with blood serotonin levels exclusively in males as well. Based on that genetic linkage finding, colleagues of mine at Vanderbilt University identified a number of rare amino acid variants of the serotonin transporter gene. Each one of these leads to an increase in serotonin transport, which potentially would lead to that increase observed in serotonin levels in the blood. The most common of these is a change from a glycine to an alanine, fairly subtle change in amino acids, but robustly associated with rigid compulsive symptoms and with sensory aversion, again, only in affected males. I should note that this variant is also found in some healthy folks, but this really enables us to transition from association studies in humans to studies that let us get at mechanism in mice. So the most common of these variants we've now studied fairly extensively in the mouse. This has now become fairly routine, where you take a particular amino acid variant, you place it in a mouse in the same location. These mice, where we've introduced these mutations, grow normally. They don't have any apparent health problems, but they allow us to do studies of the brain serotonin system to understand how this actually impacts function in a mechanistic way. So we see that it increases clearance of serotonin out of the extracellular space, and this may actually parallel taking up increased serotonin into the platelet. It increases sensitivity of serotonin receptors and decreases the firing rate of serotonergic neurons. And I could spend a lot of time talking about that, but really what we're interested in is focusing on how this may help us understand a subgroup of individuals looking at the peripheral system. So importantly, we see a recapitulation of the elevated blood serotonin levels in these mice. We also see a recapitulation of some changes in social behavior, some changes in communication, and some repetitive behavior. So you see that these animals are less prone to spend time with a social stimulus in contrast to a non-social stimulus. You see that they tend to back out of a tube when confronted with another mouse. You see that they vocalize less when separated from their mother at the postnatal day 7. And you see that they have this odd behavior where they go up and down from their cage lid over and over again, in some cases 1,000 times or more in a day. So that takes us through sort of very quickly some of the interesting things related to brain and behavior in these mice. But one of the fascinating things that we've observed is actually a change in gastrointestinal function that maybe overlaps with some findings that have been reported in autism that I know David has reviewed in some part already. But clearly there's an increase in constipation in autism, an increase in some other gastrointestinal symptoms in some studies as well. And we did a study looking at associated behaviors in autism spectrum disorder children who actually have constipation plus diarrhea or soiling. So these are really kids who have very severe constipation leading to overflow, or at least that's what we think they are. Within the ATN population, that included a strong association with repetitive behavior, compulsivity, obsessive compulsive disorder diagnosis, and so forth, suggesting potentially some overlap with what was seen in the association studies with the serotonin transporter. Interestingly, there's been a fair bit of work, mostly from Mike Gersham's lab here at Columbia, looking at mice that lack the serotonin transporter. Those mice actually have decreased transit time through their gut and show intermittent watery stool. They've been proposed as a model of irritable bowel syndrome. In our animals, we actually see something that looks to be quite opposite to that. In fact, in our animals, we see rectal prolapse in those animals allowed to live longer than a year, and increased intestinal transit time when Kara Gross and Mike Gershon have observed them. Based on that observation, they then looked for changes, and sorry, this slide has gotten a bit distorted, but changes in the neural system in the gut, and they see a dramatic decrease in numbers of neurons of a number of different classes, but particularly those neurons that we might think of as consistent with inhibitory neurons in the brain. So data that sort of bring us full circle in thinking about serotonin transporter variants associated with hyperserotonemia that are also associated with change in gut function. And that's one way that you can think about this causal pathway. Another direction is actually the opposite, to think that there might be a distortion in the way that the gut functions that could lead to elevated serotonin production, elevated serotonin levels in the blood. So the population I'm going to be talking about is actually a subpopulation of what David outlined, because not all individuals could be used because they were taking serotonin-related medications such as antidepressants that inhibit the serotonin transporter. So in this subpopulation, 82 individuals, 75 of whom were Caucasian. We did do some of the analyses in the subset. We were able to look at blood serotonin levels in relation to GI symptoms. These GI symptoms can, as David discussed, be looked at in a number of different ways. The first way we looked at it was to sort out those who clearly have a diagnosable GI disorder versus those that don't, identifying that functional constipation was clearly the most common GI diagnosis that could be made, and then identifying those individuals with functional constipation versus those that don't have a diagnosis at all. The other approach is to really think about lower GI symptoms, which are the most common symptoms seen in this population, as a quantitative score to index severity in some way. So when we look at whole blood serotonin levels, this biomarker, as a quantitative trait, we actually find that there's a skew to the distribution in the sample that requires a log transformation to get something that looks more like a normal curve. When we look at it in relation to age effects, we find that there's a significant impact of age, such that the older a child is, the lower their serotonin levels are. That's right at the threshold of statistical significance, but it's clearly something that needs to be controlled. And then when we look at blood serotonin levels in relation to historical samples, run in the same lab, but obviously some time ago, you find that roughly 24% of our population would meet criteria for hyperserotonemia. Now, we don't have actual controls in this sample, since that we were looking for relationships between this biomarker and particular symptoms. So in the primary analysis, we first looked at categorical functional constipation based on Rome 3 criteria versus no GI disorder. And initially doing the least powerful analysis, looking at the category of hyperserotonemia versus non-hyperserotonemia using the historical control. we found that there was a numerical difference, but not anywhere close to statistical significance. Then when we approach it as a quantitative analysis with regard to serotonin levels, we find that there's still not a significant relationship, but a numerical effect again, and certainly closer to statistical significance. We then did the same sort of approach from the flip side, so looking at the continuous lower GI score in the elevated serotonin group versus historical control, similar p-value, not statistically significant, but numerically in the same direction. And then the most powerful analysis looking at log transformed serotonin levels corrected for age and sex in relation to continuous lower GI score. And there we do see statistical significance right at the margin of significance and consistent on both parametric and non-parametric analysis. The R here is 0.24, so this is not a huge effect size, but right at the margin of statistical significance. We also were interested in looking at other potential behavioral variables that may relate to serotonin levels and have related in some previous studies. We saw no statistically significant relationship with self-injurious symptoms, with stereotypies, with tactile sensitivity, with IQ, only a trend relationship with compulsivity, which you recall was associated with the rare serotonin transporter variant in the population that was looked at in those linkage studies. So no clear relationship here, which honestly is quite similar to what's been seen in previous studies, where it's very difficult to pull out these behavioral variables, which perhaps makes the relationship with lower GI symptoms even more interesting, provided that it replicates in additional samples. So as always, there are lots of questions that come out. The first is this something that is real? And that really awaits replication in the second sample, and I'm hopeful that we're able to collaborate with others that are collecting these sorts of carefully ascertained samples of children with autism and GI symptoms and assess whether this relationship really holds up. Looking at GI symptoms in a quantitative way wasn't something that we really planned to do at the outset. I think it is the most powerful analysis, but we really need to understand now whether there are specific relationships with particular lower GI symptoms that may drive the correlation, and we're interested in doing that more fine-grained analysis, acknowledging that we may not have the power we'd like to have to answer that question. And then to understand, I told you that in the overall Autism Speaks to Autism Treatment Network population, there was a relationship between lower GI symptoms, specifically constipation with what we think probably reflects overflow incontinence, that was associated with particular behavioral symptoms. We haven't really looked at that yet in this population, but I think that's an area of significant interest. And then, as David, I think, was talking about at the end, thinking about ways to target one or the other measure. So we could certainly target constipation as an easy way to get started and understand whether that may change the biomarker. The converse could also be done, right? So we could use something to target the serotonin system and evaluate whether that actually changes GI symptoms. And then finally, to understand what might account for the elevated blood serotonin levels from a molecular or mechanistic standpoint. And in the mouse studies, there's some hint at that, which is that in the mice, despite all of our interest in the platelet as a particular locus where elevated serotonin transport activity could lead to elevated serotonin levels in the platelet, we actually have been unable to demonstrate that there's a clear effect on serotonin uptake in the platelet. In contrast, Kara and Mike Gershon have been able to demonstrate that there is a change in expression of tryptophan hydroxylase 1, which is responsible for the synthesis of serotonin in the enterochromatin cell, which may suggest that this is actually something where the serotonin transporter's activity in the gut leads to a change in serotonin synthesis in the gut, and then an increased release of serotonin into the peripheral circulation. We also have a hint for this in a separate population where we found that particular genotypes of the tryptophan hydroxylase locus are associated with whole blood serotonin levels, again, potentially implicating production of serotonin in the gut. Whether or not that relates to the constipation, we don't yet know. So that's my admittedly whirlwind overview, quite a number of people to acknowledge. At Vanderbilt, I would particularly like to acknowledge the work of Sarah Marler, who did all of the hands-on work with these families, walking them through a very intensive protocol. Yvonne Lee, who's been very helpful in conceptualizing the behavioral and cognitive symptoms of these children as they relate to GI symptoms. And then obviously the team at Missouri and the team at Mass General, and then a broader array of people in the lab and people who've contributed intellectually to our understanding of the serotonin system, both in these mice and in folks with autism spectrum disorder. So that's my whirlwind overview, and I'd be happy to take questions and also to engage in a discussion with David if there are questions that really bridge across multiple components of the study. Thank you very much.
We'd both also like to thank ATN and AIRP and HRSA for funding this work, as was indicated on
Thank
Dr.
Veenstra.
It was on your slide.
Yes, yes. None of this work would be possible without strong work and funding from the Autism Intervention Research Network, the ASATN, and obviously HRSA. And I will say that it's really been a pleasure to work with this team, and that the crew at Mass General has been very helpful in refining questions and providing us very helpful analyses that help us to understand what's happening in the sample.
I can't say enough good things about Aaron, Lily, and Eric.
So we actually have time for some questions. If anyone in the audience has questions that you'd like to, again, type into either the question box or raise your hand, and we can speak. But then we also have some questions from the presenters to pose to you all, and one of those was actually asked by one of the audience members, what might the treatment implications be for this set of findings? So that was posed by an audience member, and then also the presenters wanted to pose that back to the audience, if anyone has any ideas of the implications for treatment from this study.
So yeah, I can talk about that a little bit. I think one of the things that is emerging in the autism genetic and environmental risk factor literature is really quite rare risk factors that lead to a robust increase in risk. So these are things like Fragile X syndrome, found in 1% to 2% of the population, but 60% or so of folks with Fragile X syndrome would meet criteria for autism spectrum disorder, maybe a bit lower if you're narrower in the criteria. Versus the folks who actually come to clinic who usually don't have one of those rare risk factors, but may potentially share a biomarker with a population with a defined risk factor. And I would hope that something like the rare serotonin transporter variant could perhaps shed light on the larger population with this common biomarker. You might look at other biomarkers like increased rate of head and possibly body growth within the first couple of years as a parallel, but something that's pointing to a common system that if we can understand better and understand the subgroups suffering from common outcomes, we may be able to have targeted treatments that we wouldn't find a signal for in the overall population.
And on my side, I also discussed the specific implications of perhaps what abnormality in serotonin associating with, say, adequacy or inadequacy of treatment response, or association with sympathetic reactivity may have for influence of what might be suggested for supplemental treatment with either an adrenergic or serotonergic agent to help the situation.
Those are great. Thank you. Did any of the audience members want to weigh in on possible implications of treatment? I don't see any hands, so I'll go ahead. We have another question about the serotonin work. If Jeremy, if you could just discuss the relationship between brain serotonin and gut or somatic serotonin and how they might interact to affect gut function.
Sure. So the serotonin in the periphery, the serotonin in the blood, the serotonin in the gut can't cross over into the brain. There are ways that the serotonin system in the periphery clearly can have an impact. So that could be through gut function, which has an important feedback to the brain via the vagal nerve. It could be through sympathetic function, so gut distress could lead to heightened sympathetic activity, could feed back to the brain. Similarly, the brain serotonin system, you wouldn't expect to have a direct impact, but certainly could have a downstream impact on the gut. The way I usually think about these systems, and that may or may not be correct, is to think about the periphery as a model for what's happening in the brain. So if we have increased tryptophan hydroxylase activity in the periphery, maybe that same thing is happening in the brain. If we have increased serotonin transporter activity happening in the periphery, maybe that same thing is happening in the brain. And we can gain insights by working on both systems, but not that they're directly parallel. And obviously in the mouse we have some data to suggest that that's true, but I think it's really intriguing to think about interplay between the two systems, that say an abnormality in the serotonin system in the gut would lead to distress that complicates whatever is happening in the serotonin system in the brain.
Thank you. Does anyone have any other questions for our presenters? Or if anyone would like to weigh in on another question that the presenters had for you all was some other factors that might serve as biomarkers for GI problems in ASD for future studies. If anyone would like to chime in.
So I can say, and I know maybe it was part of posing that question, but one of the things I wasn't sure I'd have time to talk about is the polymorphism in the MET gene that Pat Levitt's group has looked at quite extensively in relation to gut function. And this is one where there clearly is data pointing to individuals with gut symptoms being more likely to have this particular variant as a possible risk factor for their ASD, and they point out a separate subgroup where there's really not been work yet, is trying to see whether that may overlap with this subgroup that has elevated blood serotonin levels. And you can think of ways to potentially tie the biology together that could be quite fruitful.
I'd actually contributed to that question as well with the MET gene in mind, and also possibly there are variants of the serotonin transporter that are associated with stress reactivity that one might provide, if things pan out on the parasympathetic end, that may be of relevance as well.
And one of our audience members also posed that it could be related to sleep habits or sleep patterns, and if there's any relation with high serotonin levels and those types of issues with children with ASD.
So that's a great question, and actually there's a recent paper from Thomas Bourgeron's group looking at serotonin levels and melatonin levels. And interestingly, tryptophan hydroxylase 1, which is responsible for producing the serotonin in the periphery, is responsible for, or is the rate-limiting step in producing melatonin in the pineal gland. And Bourgeron's group actually shows relationships between measures of serotonin and melatonin, and you would imagine that that might tie back together with relationships with sleep. I would say at this point we don't completely understand how those relationships might play out, but it's a very interesting area for further research.
It certainly would fit with some of the work that we've seen previously with our own registry data, where GI problems and sleep problems have tended to come hand in hand, so they sort of travel together quite frequently. It might help to tie some of this together. Very interesting stuff.
Yep. No, absolutely. I think oftentimes we write off those relationships. Write off might be not the right term, but we go for the easy explanation first. Well, you know, the child who has pain due to constipation doesn't sleep well. And it, you know, it may be that these things are actually pointing us toward common biological systems that impact those things. And only by doing these sorts of careful analyses can you start to pull those things apart.
Really is fascinating work. Wonderful work that both Dr. Beversdorf and Dr. Veenstra-VanderWeele presented today. And very pleased to see that coming out of the network and coming out of the ARP and Autism Speaks funding. So we have
wonderful. Thank you very much for hosting us.
Yes, we have come to the end of our time, but I do want to thank our speakers today for a very enlightening discussion, and hope that everybody who made it today has a wonderful Thanksgiving holiday and the rest of your week. As Audrey was mentioning earlier, thanks for attending, and you will receive an evaluation through your email, and you can complete that to receive CME credit, those of you who are interested in receiving credit. And with that, I think we'll sign off.
Yes, thanks everyone.
Thank you.
Thanks, folks.
Thank you very much, and have a happy Thanksgiving.
You too.
Thanks.
Bye now.